Thanks so much. Thanks so much.
So do you guys have an immunology specific grad program at UCLA?
No, we don't actually. We have an umbrella program through the Molecular Biology Institute that has an immunology focused home area, but it's part of a bigger umbrella group. How about Santa Barbara? Is there an immunology department there?
No, I mean, we don't have a big immunology community because, you know, we have no med school and such. Really what brought me here was the bioengineering community more than the immunology community. So we have an umbrella program that's bioengineering and molecular biology, cell biology. We have our departmental program. And then now we just started a bio-E specific program. So no immunology specific anything.
Mm-hmm. Yeah, we actually don't even have an immunology graduate course. So I'm actually going to develop one here
nice.
at UCLA. Yeah. So we're definitely.
I'm actually not sure what we have. I mean, most of our grad courses are like pretty focused on the things that truly everybody needs to know, like how to read papers and the basics of like how gene cells work.
Yeah, likewise, and writing grants and other proposals.
Yeah.
Let's go ahead and get started. It is my great pleasure to introduce Meghan Morrissey today. She's currently an assistant professor at University of California at Santa Barbara and has been there since 2020. So has really only experienced, I guess, PI ship in the pandemic world. Meghan trained at the University of Notre Dame and received a BS in biology and was in their honors program and finished magna cum laude. And she had a thesis on building a platform for kinetochore dynamics. She then went on to do her PhD at Duke University in genetics and genomics, as well as cellular and molecular biology, under the advisorship of David Sherwood. She then moved to California at University of San Francisco, where she did her postdoc under the advisorship of Ronald Vail, studying the mechanisms of phagocytic signaling. And she's going to share some of the really exciting work that her lab has been doing on engineering phagocytosis. Thanks so much, Meghan.
Thanks for having me. I'm really excited to be here. Can everyone hear me? Yeah, okay, great. Yeah, so as you may have gathered from that introduction, I am really a cell biologist at heart. I just think that immune cells are some of the coolest cells with some of the most interesting challenges. And so that's kind of the perspective that I take on all of my research. So I think probably everybody here knows that immunotherapy has been a huge recent advance in cancer therapy. And while it's been revolutionary, I think that it's not reaching as many patients as it could right now. So for example, T-cell checkpoint blockades, the most notable success story, only work in about 25% of patients. So I think as immunologists, it's really important to start thinking about how we can engage other cell types in the cancer immune response. And so one problem with everything that we're doing right now is the vast majority of it targets T cells. And by targeting T cells, that's really going to limit things just based on the basic biology of T cells. So for example, T cells are maybe not the best cells at getting into solid tumors. And so in contrast, macrophages, the cell.mylab studies, are specifically recruited to solid tumors. And once macrophages are in solid tumors, they can do a lot of different things that can either promote cancer growth or cancer destruction. So for example, macrophages can phagocytose cancer cells. They can secrete cytotoxic proteins to directly kill the cancer cells, or they can stimulate T cells so that the T cells are more able to enter the solid tumors and more able to kill the cancer cells. But despite all of these really great things that macrophages can do, the net effect of macrophages in the tumor is negative. So more macrophages correlates with poor patient prognosis. And that's because macrophages also do some really bad things. They can promote cancer metastasis, and probably most critically, they can contribute to this immunosuppressive microenvironment that keeps T cells from functioning in the solid tumor. And so, like I said, my lab, we're really cell biologists and synthetic biologists, and we want to understand and rewire all of the signaling pathways in macrophages that are currently being targeted by cancer immunotherapy. Now, that's kind of like the big picture of all of the things that my lab is interested in and why we're interested in them. But zooming in on one specific problem, today I'm going to tell you about how a macrophage decides what to eat. And at first glance, if you read a review article, it might seem sort of simple. Macrophages will recognize things called eat me signals. And these eat me signals say, eat me. This is a good target to phagocytose. So one that I'm showing here is IgG antibodies bound to this pathogen. But if you think about it a little bit harder, of course, it's not so simple. And one of the problems is that eat me signals are constantly present in healthy tissues, sometimes at quite high concentrations. So here I'm showing just IgG antibodies, one of many eat me signals that macrophages can recognize. And it's present at mg per mil concentration in healthy adult plasma. So that's a lot of protein. And in the face of that super high background, even healthy cells like these red blood cells are going to transiently or non-specifically bind to some eat me signals. And so it's very important that macrophages rapidly clear pathogen and debris, but it's equally important that they robustly ignore healthy cells, which there's many more healthy cells in a tissue than pathogens, so this can be quite a challenge. So how does a macrophage do this? Well, there's kind of two sides to this. First, they measure the amount of eat me signals. So it's not simply the presence of a few eat me signals that can activate a macrophage, but it has to be enough to trigger some sort of reaction. And then second, they will recognize don't eat me signals on healthy cells. And the most well-known of these is CD47. So how does a macrophage measure and add all of the different signals on a target to decide whether or not it should eat it? Today, I'll tell you two parts of my talk. So part one, focusing on how macrophages measure eat me signals. And then part two, focusing on how the don't eat me signals prevent phagocytosis. Now, I did say that we were interested in this because of cancer immunotherapies. And so the reason I got into this whole pathway is that CD47 is upregulated on cancer cells. And it's thought that this allows them to hide from the innate immune system. And so for this reason, there's many different CD47 blocking strategies currently in clinical trials. And they can be administered alone, but more commonly, they're administered with therapeutic antibodies as a combination therapy. And so this basically means you can add an eat me signal to the cancer cells while also inhibiting the don't eat me signal to allow for these cells to be attacked. Okay, so if you want to quantitatively study how a macrophage measures different signals, it's really important that you know quantitatively what signals the macrophage is encountering. And in vivo, this can be kind of difficult because every target that the macrophage encounters will be different. It can vary in size, shape, stiffness, and most problematically for us, it will have many different signals on its surface. And somehow it's the combined action of all of these signals that will tell a macrophage eat or don't eat. And while I'd love to eventually understand all of this complexity, without being able to control exactly what amount of signals are there, or even knowing the identity of all of the signals on a target, it's very difficult to make progress. And so to start with, I'm using a reconstituted target where I can control exactly what signals are there, exactly what densities, and as you'll see later in my talk, even sometimes the pattern. Okay, so what does this target look like? Let's zoom in on this interface. So I start with a glass bead, and on that bead, I build a supported lipid bilayer. So this mimics a cancer cell surface. It's mobile, proteins can move around just like in a cell membrane. This bilayer contains a biotinylated lipid, and it's recognized by antibiotin IgG. This activates the Fc receptor in the macrophage, which then signals for phagocytosis or engulfment. And so our go-to assay is pretty simple. We make these reconstituted targets, these beads that you can see in green. We incubate them with macrophages, and then we use high content image acquisition and automated analysis pipelines to count how many beads the macrophage has eaten. And so here I've done a pretty simple experiment where I've just titrated the amount of IgG bound to my targets. And you can see when there's a low level of IgG, the macrophages mostly ignore the targets. But when there's more IgG, now they are phagocytosed. Okay, so going back to this problem that I posed at the beginning, I think it's pretty obvious that if a single IgG binding to a single Fc receptor was sufficient to activate phagocytosis, that would be a total disaster and healthy cells would get eaten all the time. And so phagocytosis requires some critical threshold of receptor ligand interactions in order to occur. But what is this threshold? How is it set? How does the macrophage measure the amount of IgG on a target surface? So this question was really interesting to me, and it was really interesting to Nadia Kern, who's a graduate student at UCSF. We worked together while I was at UCSF, and then while I was starting my lab here at UCSB. Okay, so we had some clues about how a macrophage might measure IgG that came from turf microscopy. So one of the nice things about our system is you can take exactly the supported lipid bilayer that you built on a three-dimensional target and flatten it out on a two-dimensional glass cover slip. And then you can use turf microscopy to illuminate a very thin slice of the macrophage right where it's interacting with the cover slip and get really high resolution images of the molecular dynamics as this macrophage tries to eat this giant target, which will obviously never be able to phagocytose. Okay, so when you do that, this is the sort of data that you get. So here I'm showing a turf movie of IgG on a supported lipid bilayer. And at the beginning of the movie, the IgG, which is in black, is diffusing around the cover slip rapidly, so it looks kind of gray, blurred out. And during this movie, you'll see a single macrophage land. And as this macrophage lands and spreads across the surface, the IgG is reorganized into these nanoscale clusters. We know from previous research that these clusters are hotspots of active signaling. It's where the downstream signaling molecules are recruited and activated. And so we thought that this was really interesting and might have something to do with how macrophages measure IgG. But the problem previously in studying these clusters is that it's very difficult to manipulate very tiny things like this on the nanoscale. But we thought that maybe we had a series of questions we wanted to address. So first, does receptor clustering affect engulfment? Second, what constitutes a cluster? This is kind of a vague term, but how many molecules does that mean and what sort of spacing? And then finally, the thing that we were most interested in is why are clustered ligands more potent? Why is it not just the number of ligands regardless of the position? So there were a couple of things that put us in the right place at the right time to start to address how these clusters might contribute to signaling. And so one of them was that we had some colleagues at UCSF who were using DNA origami to pattern T-cell ligands. So Sean Douglas is a DNA origami expert, and then Ray Dong was our colleague in the Vail lab who was using these structures to study T-cell signaling. And so Sean and his lab designed this DNA origami pegboard. So there's biotins on the bottom to attach it to our supported lipid bilayer. There's fluorophores on the corner so that we can track it. And then most importantly, there's 72 independently modifiable positions on the top where you can either expose the ligand or not. And so this would allow us to generate clusters that were about the same size as that movie I just showed you. So this pegboard is 60 nanometers by 60 nanometers. Our normal phagocytosis targets are 5 microns, so you can put many, many, many pegboards on a bead. Okay, the other thing that positioned us to do this is that I had just finished a project designing chimeric antigen receptors that triggered phagocytosis. And these were based on the Fc receptor. And what I found out during this study is that you can replace the extracellular Fc receptor antibody binding interaction with basically anything that recruits the intracellular ITAMs of the Fc gamma chains to the cell target synapse. And so Nadia designed a chimeric antigen receptor that uses DNA hybridization to activate the receptor. So basically she replaced this interaction with DNA. And she did a lot of things to characterize this receptor and show that it signals the same way as the native receptor. But I think the most important is this simple piece of data. The DNA receptor triggered phagocytosis just as well as the endogenous system. And so with this tool, we were then able to combine this DNA CAR with this DNA origami pegboard to ask, how does manipulating the spacing between receptors affect their signaling? Okay, so back to our questions. Our first question is, does receptor clustering affect phagocytosis? And so Nadia did this experiment. She made DNA origami pegboards with different numbers of ligands, so 2, 4, 8, 16. And then what you really want to do this experiment well is to have beads that have the same number of pegboards, the same number of ligands, but you've only changed the spacing. And so she diluted all of these out with the appropriate number of these blank origamis. So the surface of the bead would look something like these pictures. And so here's the data she got, just measuring phagocytosis. And already you can see that, yes, clustering matters. It enhances phagocytosis. And already you can see that we're starting to answer this question: what constitutes a cluster? So Nadia found that maximum phagocytosis happened when there were clusters of 8 ligands or more. And that 4 was really almost just as good, but 2 was significantly worse. And so we honed in on this 4 ligand cluster to start manipulating the spacing to see if we could define what sort of spacing was required. So she did this experiment where she built 3 different types of pegboards, one where the 4 ligands are tightly clustered in the center, one where they're a little more spaced, and then one where they're maximally spaced, and asked, how does this change in spacing affect phagocytosis? And she found that these tightly clustered ligands were about twice as good as these more spaced apart ligands. So this really drives home, yes, clustering matters. And that cluster has to be quite tightly clustered in order to benefit from clustering. Okay, but the real reason we were excited about this data was that now we have a tool to ask, why are clustered ligands more potent, which was kind of the fundamental question we had all along. And so we took a pretty simple approach. We made orange beads covered in tightly clustered ligands, and we made blue beads covered in spaced apart ligands. Then we fed these beads to the same macrophages, and we just used time-lapse microscopy to watch phagocytosis and see what happens. What you can probably see from this movie is the orange beads are phagocytosed much more. But if you break down all of the different steps, the macrophage is able to interact with both types of beads. It binds equally well to the blue and the orange beads. But it's much more likely to initiate phagocytosis of the orange beads. However, if it does initiate phagocytosis of the blue beads, it happens at about the same rate as the orange beads. So this really pinpointed to us this initiation step in starting phagocytosis or triggering the process. So given that, we really honed in on the very first few molecular events that are required for phagocytosis. So the first thing that's required is receptor binding. And we did a number of experiments that basically convinced us that receptor binding was not affected by clustering. And so here I'm just showing you one kind of straightforward experiment where we have a macrophage that's encountered these orange beads with tightly clustered ligands or the blue beads with spaced ligands. And we didn't see any difference in receptor recruitment to these different beads. So the next thing that happens is the receptor is phosphorylated. And we measured receptor phosphorylation using the tandem SH2 domains of SYK. So SYK is a kinase and a complicated protein with lots of regulatory events, but this tandem SH2 sensor is very inert, and it has a really rapid on-rate phosphorylated receptor that we've measured ex vivo. And we found that already there was a really big difference in tightly clustered ligands unspaced. So much more receptor phosphorylation when the beads had clustered ligands than when they had spaced ligands. We continued to see this difference throughout the rest of the signaling pathway. So there was also more PIP3 generation and actin filament generation when the ligands were clustered. And so all of this really pinpoints receptor clustering affecting receptor phosphorylation. And I think it's kind of interesting to start to think about why just changing the nanoscale localization of these receptors might have such a dramatic effect on the kinetics of this process. Okay, but we didn't solve that question. So I just want to step back and say, how does this relate to the question I initially posed? How does a macrophage measure IgG? So what we imagine is that, if you're a macrophage navigating a complex tissue, you do not have time to scan every single cell to figure out how many IgGs are bound across the whole surface. But this mechanism suggests that perhaps if a macrophage touches a cell and it's got an IgG molecule, but it doesn't find any IgG molecules nearby, that's a pretty good indication to ignore the target. However, if it does encounter one IgG and there's more nearby, a cluster, that's a pretty good indication that this is a good target for phagocytosis and the macrophage should initiate signaling. I think it's also interesting to think about Nadia's study in design of therapeutic antibodies. So there are some therapeutic antibodies that trigger antibody-dependent cellular phagocytosis. I think rituximab is the canonical one of these. And then there's others that, to our surprise, rely on Fc receptor activation for at least part of their function. And that's the rest of the antibodies on this list. So we know some parameters that make for effective antibodies. For example, antigen affinity, I think pretty obviously, antibody isotype, and antibody glycosylation. And we're starting to learn more about the biophysical parameters that may also affect antibody signaling. So for example, antigen height, we now know can impact how well an antibody signals. And then I think that Nadia's study really suggests that antigen pattern also can affect how well an antibody signals. Okay, so after Nadia was done with her study, we still had a lot of questions about how a macrophage can measure IgG that we couldn't really address well with our techniques that we had. So one problem with using beads is that you can't control when beads contact a macrophage. So you can't control when or how many will touch any individual cell. And if you want to ask questions about how a macrophage responds to a really carefully controlled dose of IgG and whether a high versus low dose is different, that's difficult. So Annelies Bond was the very first rotation student in my new lab here at UCSB. And for her final rotation, she decided to start a collaboration with Max Wilson, who's an optogenetics expert on campus. And they thought it would be fun to design an optogenetically controlled Fc receptor so that we could start asking these questions about dose. And so the construct that Annelies designed is like this. It has an intracellular ITAM and then a fluorophore and Cry2. So when Cry2 is exposed to blue light, it undergoes a conformational change and it homoligamizes. And so Annelies thought that based on Nadia's work that clustering is so important, this might be enough to activate clustering and initiate phagocytosis. And obviously I wouldn't be telling you this if that weren't the case. So here's a movie that Annelies took. This is a macrophage and the optogenetic construct is shown in green. And then these beads in pink, they don't have any IgG. They just have a little ICAM so that they stick to the macrophage better. And so what Annelies did then is shine blue light on them, and this induces cluster formation. And you can see the macrophage immediately starts phagocytosing these beads. So just quantifying that over a few different experiments here. Okay, so I'm just going to show you one piece of data that Annalise has from this construct so far. So she wanted to know how previous encounters with IgG-coated particles or previous phagocytic events would affect future phagocytic events. So how does a light-based appetizer affect macrophage eating its whole meal? So what she did is stimulate macrophages for 15 minutes with light and then stop. And the clustering of the optogenetic receptor is reversible, so it'll totally disassemble and be inactive. She waits 1 to 4 hours, and then she re-challenges the macrophages with IgG-coated beads and allows phagocytosis to happen. And so when she does this, if you just feed the macrophage IgG-coated beads, they eat them, of course, compared to the controls. But if you have previously stimulated the macrophages, now they eat twice as many beads. And this enhancement is true at 1 hour and also true at 4 hours. So basically, Annalise has a lot of experiments to do. She wants to know what the molecular memory that macrophages hold for these past encounters with IgG is. What's the mechanism for this? Then we also want to use this as a tool to dissect Fc receptor signaling further to ask whether different doses elicit different responses. And then we also think that this might give us some insight in how we can engineer hungrier macrophages that might be useful for therapeutic things like therapies that induce antibody-dependent cellular phagocytosis or CAR macrophages. Okay, so that's all I'm going to tell you about how macrophages measure IgG. And now I'm going to move on to telling you a little bit about how CD47 prevents phagocytosis. Okay, so to study CD47, we really need to add it into our reconstituted system. And so we do this by adding the purified extracellular domain to our targets. This CD47 is recognized by SIRPα in the macrophage. When SIRPα binds to CD47, it gets phosphorylated, it has intracellular ITIMs, and recruits SHP1 and SHP2 phosphatase. So first, we had despite really intense therapeutic interest in this signaling pathway, we thought that the basic biology, there's still a lot of really open, big questions. So first, how is SIRPα's activity controlled? How does CD47 binding activate SIRPα? And when CD47 isn't there, how do you prevent SIRPα from being activated? And then second, what are the targets of CD47 SIRPα signaling? So once this phosphatase is recruited, what does it actually do to prevent phagocytosis from happening? Okay, so first let me just show you that I'm going to first tell you about this first question and suggest that SIRPα's activity is controlled primarily by its localization. Okay, now let me show you that this works in our system. So if you have beads with IgG, and then you add CD47, you see much fewer beads phagocytosed. So CD47 is inhibiting this process. Okay, so when I started this project, one of the very first things that I noticed is that when macrophages encounter IgG-coated beads, so no CD47, these are beads that should be phagocytosed, SIRPα is depleted from the synapse between this macrophage and the bead, or the phagocytic cap. However, when CD47 is present, now this depletion no longer occurs. And I thought this was really interesting based on what we know about the phagocytic synapse and how signaling happens there. So here's what we know. When IgG binds to the Fc receptor, it brings the macrophage cell membrane and the target membrane very close together. And this small intermembrane space drives steric exclusion of these bulky phosphatases. There's basically not room for them to enter this area. These phosphatases would normally keep the Fc receptor inactive. In contrast, the kinases, the SRC family kinases that activate the Fc receptor are present throughout the plasma membrane. And so with the bulky phosphatases out of the way, this creates a kinase-rich zone that favors receptor phosphorylation and activation, and a phosphatase-rich zone at the periphery where things are not activated. And so the data that I just showed you suggests that when CD47 is absent, SIRPα is in this phosphatase-rich periphery. But when CD47 is present, now this reorganizes SIRPα, brings it into the kinase-rich zone. And I wondered if this simple change in localization was sufficient to activate SIRPα. And so to do this, to test this, I did a number of different things that changed SIRPA's localization in the absence of CD47, but I'll just tell you about one. So I replaced the SIRPα extracellular domain with an antibody binding extracellular domain. So now it will localize to the synapse exactly the same way the Fc receptors do. First, let me show you that that's true. So here is a macrophage encountering IgG-coated beads. The full-length SIRPα is depleted here, but this chimeric protein is not, it's enriched. And this does suppress phagocytosis. So this suggests to me that simply by bringing SIRPα's intracellular domain into this area where it can be phosphorylated, that's sufficient to activate the signaling. Okay. So next I wanted to know what drives SIRPα's exclusion from the synapse. Thinking about it, it seems really important to keep SIRPα from just diffusing into this zone where it can be activated. Because if SIRPα could just diffuse into this kinase-rich zone, it would be like driving with the brakes on. And so I wondered if perhaps SIRPα's extracellular domain was bulky enough to drive steric exclusion, just like these phosphatases that I'm talking about. So the protein component of SIRPα is not so tall, but it is heavily glycosylated. And so I basically decided there's two possibilities. First, SIRPα's exclusion could be driven by some sort of signaling event induced by IgG, or SIRPα's exclusion could be induced simply by this binding event and the formation of this sterically confined synapse. And so I decided to design a bunch of synthetic tethers that would alter the distance between the macrophage and the bead without activating phagocytosis or without inducing any sort of Fc receptor signaling cascade. And so the tethers I built look like this. On the tip of each tether, there's FRB, and on my beads, there's FKBP. And so these proteins will form a chemically induced dimer in the presence of rapamycin or a rapamycin analog. Then the spacing of these proteins is controlled by a differing number of FibCon repeats. So I have one with 0, 1, 3, 5. And basically when you stack these FibCon repeats on top of each other, it makes a semi-rigid protein. So kind of like a pool noodle compared to like a tent post. And so in this way, I could alter the spacing between the macrophage and the bead and ask how SIRPα exclusion was affected. And when I did this, I found that short tethers, similar in size or smaller to the IgG Fc receptor interaction, were able to drive SIRPα exclusion from the synapse, but long tethers were not. So this really convinced me that SIRPα is sterically excluded from the phagocytic synapse. And so the way that I'm thinking about this is when the Fc receptor is bound to IgG, it's energetically unfavorable for SIRPα to diffuse into the synapse. However, when CD47 is present, that ligation provides sufficient energy to retain SIRPα at the synapse where it can be activated and suppress phagocytosis. Okay, so that's all I'm going to say about how SIRPα's activity is controlled. Next, I'm going to tell you about what the targets of CD47 SIRPα signaling might be. And I'm going to suggest that at least one major target is integrin. Okay, so when I started this project, I wanted to use turf microscopy to look at the molecular dynamics at the phagocytic synapse when CD47 was present or absent. And so you might remember this movie from earlier in the talk. It's one macrophage landing on an IgG-coated bilayer. And as the macrophage lands, it forms these nanoclusters of IgG. These recruit downstream signaling molecules. So the first thing I did was see if there was any difference in this process when CD47 was present. And I tried really hard, looked a lot, and really did not see a major difference here. So SIC was still recruited, the clusters still formed, and when I quantified these, I didn't see a difference. However, when I was doing these experiments, there was something that was dramatically different. So let me just play these movies. So here, a macrophage is encountering an IgG-coated bilayer, and here the macrophage is encountering the same bilayer, but also with CD47. And you can see that this macrophage, it lands and it spreads, it's very elegant and coordinated. But this macrophage is kind of uncertain, and the way it interacts with the bilayer is quite different, like a ice skater that's just learning how to navigate the ice. So I quantified this over many macrophages, When a macrophage lands on an IgG-coated bilayer, it spreads across the surface, but when CD47 is present, that's really inhibited. And while the spreading is not phagocytosis, I thought that it might be a clue for what CD47 was doing to prevent phagocytosis. And one molecule that really stood out to me to look at was integrin. So integrin is required for many different cellular processes: cell migration, cell adhesion, cell polarity, and it's also required for phagocytosis. So when IgG binds to the Fc receptor, that triggers many different intracellular reactions, including inside-out activation of integrins. So what do I mean by inside-out activation of integrin? Well, normally integrin exists in the plasma membrane in this low affinity bent conformation. But when it receives an inside-out activation signal, it flips open into this open conformation where it can interact with more ligands at much higher affinity and also serve as an intracellular signaling platform, for example, for actin assembly. So the first thing I needed to do was test if integrins were required in my system, because my bilayer doesn't have any specific ligands for them. And so I did that. I treated with a beta-2 function blocking antibody to eliminate integrin. When I did that, phagocytosis was significantly decreased. So integrins are required for phagocytosis. The next thing I wanted to do was see if CD47 changed integrin activation. And so to do that, I stained for a marker of active integrins, phosphopaxillin. And when macrophages encounter IgG-coated beads, phosphopaxillin's enriched at this phagocytic cup. But when CD47 is present, it's not enriched. And this was in line with previous studies from Dennis Disher's lab. So we were pretty convinced that CD47 inhibits integrin activation. Okay, so at this point, my model looked like this. The Fc receptor activates many things that culminate in phagocytosis. And I thought that SIRPα prevented integrin activation. However, it's also possible that SIRPα could act on a more upstream target, or it could act somewhere in this pathway as well. And so I reasoned that if this model was correct, if the main thing that SIRPα was doing was preventing inside-out activation of integrins, then I should be able to bypass the suppressive effect of CD47 by providing an outside-in form of integrin activation instead. And so I did this in two different ways, both of which lock integrin into this high affinity open conformation. So a surplus of ICAM, which is a high affinity ligand for integrin, or manganese, which is a divalent cation. And when I did this, it did bypass CD47, totally eliminated the effect. So here's the data. When you feed IgG-coated beads but add CD47, this inhibits phagocytosis. However, if there's manganese present or ICAM on the beads, now CD47 has no effect. So all of this together suggests that activating integrin bypasses CD47 signaling. I think this is like a pretty big step forward in understanding how CD47 works, because we really would like to know how it prevents phagocytosis. And this suggests that whatever the direct targets of SHP1 and SHP2, the SIRPα bound phosphatases are, the main thing they're doing is preventing integrin activation. Now, recall that I am interested in this problem because of its cancer relevance. So if activating integrins can bypass the suppressive CD47 signal on beads, can it also bypass the suppressive CD47 signal on cancer cells? And so to test this, I incubated macrophages with cancer cells. And so I've labeled here just the nucleus of the cancer cell. So it just looks like a blob with the unlabeled cytoplasm around it. And normally, if you add cancer cells to macrophages, the macrophages ignore them. But if you activate integrins, now you can see this macrophage reach out and phagocytose that cancer cell. Quantified over many time lapses, activating integrins worked about as well as the CD47 blockade. So this suggests that it does bypass the CD47 suppressive signal. Okay, so to sum up my model for how CD47 works, If you do not have CD47 and only have IgG, SIRPα can't enter the phagocytic synapse, so it's not activated, and it's also not positioned near its downstream targets. However, if CD47 is present, now SIRPα can enter this kinase-rich zone to be phosphorylated, and it may also position SIRPα near its downstream targets so that it can better inactivate the signaling pathway. And I think this also, thinking about it more, it makes a lot of sense because integrin is involved in many different types of phagocytosis. And we also know that CD47 is a pretty broad inhibitor of phagocytosis. So no matter how you're activating phagocytosis, it's possible that this mechanism could hold true for all different types of phagocytosis pathways. Okay, now let me just tell you one short vignette about CD47 in my own lab. So everything that I have told you so far, I've been showing you this pathogen that has IgG on it and clearly doesn't have any CD47. But what if the macrophage is trying to phagocytose a cell? What if that cell previously had CD47 on it? And so this happens all the time in our bodies when cells undergo apoptosis. So apoptotic cells are cleared so rapidly from tissues that you hardly ever find an apoptotic corpse in a tissue at homeostasis. And so how do apoptotic cells get phagocytosed? Well, we know that they upregulate eat-me signals, and the most well-described of these is phosphatidylserine. We also know that they inactivate CD47. And we know this because when you incubate apoptotic corpses with macrophages, you don't get any phosphorylation of SIRPα, but if you incubate viable cells with macrophages, you do. And so how CD47 is inactivated is a bit of a mystery that Wyatt Miller in my lab is trying to solve. So we got interested in this because you could imagine the simplest scenario is that CD47 is simply downregulated from the cell surface. But that doesn't appear to be the case. And this paper described that in really nice detail, but we wanted to do the experiment in our own hands. So we induced apoptosis by adding hydrogen peroxide to the macrophages, and then we measured how much CD47 was present to see if it was downregulated or internalized. And we did not see any change in CD47 levels upon induction of apoptosis. So here we're just comparing viable cells and apoptotic cells that have been treated with hydrogen peroxide and exposed phosphatidylserine. So something else is happening to inactivate CD47, and we would really like to know what. So the first thing that we did was to use CRISPR to tag the endogenous CD47 locus with GFP. And so when we imaged this GFP tagged CD47, we saw that it looked quite clustered or punctate within the plasma membrane. So this is CD47 GFP compared to a membrane dye or a membrane tethered protein. You can see it's different. Okay, so this is interesting, and it was especially interesting to us once we induced apoptosis and looked again. In this case, we saw that those clusters of CD47 that we saw in the previous image had disappeared. So here we have CD47, again, GFP tag, compared to a membrane dye or a membrane tethered protein. And while there's dramatic changes in the plasma membrane, the clustering of CD47 is clearly gone. So here's just showing those side by side. Now, to quantify this, we thought, okay, well, if CD47 is uniformly distributed across the plasma membrane, then a membrane dye should predict CD47's intensity quite well. And that is indeed the case for just a generic membrane tethered protein, a CAXM cherry. So the CAXM cherry correlates quite well with a membrane tethered dye on healthy or apoptotic cells. However, CD47 only correlates with this dye well on apoptotic cells, indicating that it's just diffused throughout the membrane. But on healthy cells, that correlation is much different. So this really suggests there's a change in the organization of CD47 upon apoptosis. So our hypothesis is that when CD47 is on healthy cells, it's clustered, and maybe this inhibits phagocytosis. However, when a cell undergoes apoptosis, it becomes dispersed, and we hypothesize that then this is less able to inhibit phagocytosis. And so Wyatt now is using a lot of synthetic biology techniques, including the DNA origami that Nadia developed, to manipulate CD47's pattern and test these hypotheses. Okay, so that is all I'm going to tell you about how macrophages select their targets for phagocytosis. I just want to end by thanking my lab. So this picture is from a year ago. We already have some more lab members. Thanking Nadia and our collaborators at UCSF and UCSB, and then also saying that we're hiring at all levels. The Morrissey lab is especially looking for postdocs, and then my department here at UCSB is hiring an assistant professor, and the application is due pretty soon, I think next week. So come join us. It's a beautiful place, as you can see from my picture right here. Our building is right here. So thank you.
Meghan, that was fantastic. Really amazing work that you've been able to accomplish in 2 years and during a pandemic. So exciting. If anyone has questions, feel free to put them in the YouTube chat, and I'll read those. And I guess I can start. How do you, obviously you sort of started from the cancer world, right, of trying to design something that we can attack the cancer cells, right? So how do you envision this being used?
Yeah, so for the for each project, it's a little bit different. I think that, what we're mostly interested in is understanding how these pathways work. And we don't always have a plan for exactly what we're going to find, because that's not really how it works. But for Nadia's project, for example, I could envision that really informing which targets are good targets for antibodies. So if you're designing a therapeutic antibody and it's a protein that's not diffusible within the plasma membrane, maybe it's not going to signal as well as something that's perhaps like localized to clusters. That would maybe be a more potent target. And then for Annalise's work, we're really interested in how these, how we can use what we've found to engineer hungrier macrophages. So macrophages do antibody-dependent cellular phagocytosis. This is the mechanism for many monoclonal antibodies. Then there's also a number of biotech startups designing chimeric antigen receptor macrophages. And those reportedly function through phagocytosing cancer cells, as well as probably secreting some inflammatory cytokines and stuff like that. But if we understood how to enhance their signaling, how to make them hungrier, that could be really interesting. Yeah, and then for CD47, I guess for Wyatt's project, what we're really thinking about is there's a lot of different times where you want CD47 to be less potent, that's cancer. But actually, this is probably more relevant to the times where you want CD47 to be more potent. For example, anytime you're putting something into a body and you want to protect it. So if you're putting stem cells into a body, often people will overexpress CD47. If you're putting in nanoparticles, often they'll be coded in CD47. I think it's interesting to think about how we can make those more potent.
Yeah, absolutely.
Yeah, it's really awesome talking, Meghan. I really like all the mechanistic work you're doing and all the really cool technology you're kind of developing in your lab. It's really awesome. So I study aging and senescence. And so I think it's really interesting that, senescence is becoming it was emerging as a driver of inflammation associated with aging and a driver of lots of aging-related diseases. And it's thought that it's speculated that perhaps senescent cells undergo avoidance or of phagocytosis by immune cells such as macrophages or clearance by NK cells. So I was interested to see if your thoughts about maybe looking at the role of CD47 potentially in senescence and how they potentially may avoid phagocytosis by macrophages, or if you thought about that.
Yeah, I think that's a super, super interesting question. So there are a few studies looking at CD47 during aging, and I certainly don't think it's a solved question, but there is some literature there. But I think kind of more broadly, it's clear that there's different times where macrophages' phagocytic capacity is different. So I think like my favorite example of this is in the CD47 knockout mice, macrophages don't just eat everything. So somehow during their development, they tune how much phagocytosis they should be doing based on the absence of CD47 everywhere. But in contrast, if you have a wild type mouse and you add in CD47 negative cells, they get cleared really fast. So I think like how macrophages tune their sensitivity is this basic idea that I'm hoping our studies can start to uncover. That's super relevant for aging and for all sorts of different scenarios. Yeah, and then I think, like you say, there's kind of the other side too, which is how do the targets protect themselves? So there's like, how do the macrophages change? And then how do the targets change? Yeah, all really interesting.
Awesome, thank you.
Megan, do you think this is different in sort of sites of active inflammation when there's just so much more antibody around, right?
Yeah, totally. So we are kind of just modeling, not that, like a really basic context so that we can get a handle on the problem. But yeah, I expect that kind of all of the like levers that macrophages and cancer cells can pull to change phagocytosis or something, those are probably going to be pulled during inflammation because that just makes sense. And so, yeah, first we identify the basic mechanisms and then we see how they're changed in inflammation. But yeah, I totally expect it to be different and interesting.
Doesn't look like there's any other questions, so maybe we'll go ahead and end the live streaming, Megan, and you can stay in the Zoom.
Okay, great. I'm gonna stop sharing.
Perfect. Sorry for the, and there's something with my reset computer that I couldn't start and stop the YouTube link, even though I like it. So
Yeah.
Shu had to do it from wherever he was. So yeah, no, it's super, super cool stuff.
Thanks for having me. It's always fun to present to immunologists because, you know, like I said, we're thinking about the cell biology of the questions. And so then the questions I get from like cell biologists and biophysicists and immunologists are like totally different and it's fun. Yeah, but I really like your aging idea. I've thought about like long-term how that would be an interesting project, especially because as I am sure you know, there's like, well, as a new PI, I've already gotten funding from NIGMS. So now I have to get it from a different institute. And the National Institute of Aging would be awesome.
Yeah, I think there's definitely tons of opportunity, to apply your